Hello to all of you. I'm going to give you some instructions for the labs you are going to do this week. So basically what you have to do these labs at home, and I have created a folder on the Mercer canvas which says activity oblique observation folder. I want you to perform the lab at home, and then there are like some spaces for microscopy lab. At the end you can see there are some circles. So basically in this lab you are going to go over some of the pre-made slides which have been given to you with the kit. So you are going to observe those slides under the microscope, and you are going to make some pictures here on these pages, and you're going to click the pictures and you have to upload on the Mercer canvas. So that's not a graded part, but I want to make sure that you guys are able to perform the labs at home. I want to see that. But the graded part will be the quizzes. So I have created post-lab quiz. So those quizzes you can answer only once you have done the lab at home. Okay. So microscopy, last week I have posted one link which is a virtual lab for microscopy. So it gives you a good idea about how to use a microscope, basic information about the microscope, and basically in this lab you have to just watch some of the pre-made slides which you have got along with the kit. So basically, as you know that the microscopes, why do we use the microscope? So microscopes we use to see something which is very small in size, which we just cannot see with our eyes. So this is a microscope which you have received in your kit. And there are two kinds of microscopes. One is a stereo microscopes, dissecting microscopes, and the second kind of a microscope is called the compound light microscope. And the stereo microscopes, they are used to view large objects such as fruit flies. Compound light microscopes, they are used to view the small things. Okay, before I go and talk to you about the different parts of the microscope and how to use the microscope, I would like to introduce some of the terms which are important for you to know. First of all, what is resolution? So resolution is the ability to distinguish between two adjacent objects. The lower the resolution of an image, the blurrier it appears. Okay, or the image is blurry. Now the next term which you need to know is depth of field. So depth of field is the term which is used basically when you focus some object. So how deep image you can see, that is the depth of field. So how much deep sample you can see when the sample is in focus, that is the depth of field. And depth of field decreases as the magnification increases. Next term which you need to know is parfocal. So parfocal is a term which is used basically when you have some image which is once you have some like some sample on your slide, you focus it. So when you change the magnification, suppose you have focused something at 4x, okay, and then you change to 10x. So basically the image stays in focus, okay. When you change from 4x to 10x, the image usually stays in focus, but you have to just do some fine focusing. That is what is meant by the parfocal. Yeah. So parfocal we can also define as if the image is focused. If the image is focused, as I said, it should remain in focus when you change the objective with minimal fine tuning using the fine adjustment knobs. Okay, now let's talk about the structure of the microscope. This is the microscope. Okay, so there are different kinds of light microscopes. Some microscopes have one eyepiece, some have two. So there is one eyepiece. Those kind of microscopes are called monocular microscopes, and some of the microscopes, compound light microscopes, they can have two eyepieces also. Like this is the eyepiece. Eyepiece means this is the lens through which you see your magnified image. So some of the light microscopes, they can also have two eyepieces. So those kind of microscopes, that binocular microscope. So this is the basic microscope. So this is the eyepiece, and this is the nose piece, and in the nose piece there are different kinds of objective lenses. Now why they called objective lenses? Because these lenses are near the object which is on the slide. And these objective lenses, they are of different magnifications. They can be of 4x, 10x, 40x, and 100x. Okay, so this is the stage, and on the stage there are stage clips in which we insert our slide, and on the slide we put our sample, and we cover our sample with the cover slip. And this is the coarse adjustment knob, and this is the fine adjustment knob here. Okay, and this is the light source. From here the light comes. This is the diaphragm. So this diaphragm can be adjusted, and through this diaphragm the intensity of light can be adjusted. You can open it, you can close it. By closing it, you basically restrict the light which is going through. So the light is coming from here, and then here is a condenser lens. The light gets condensed here, and then here is your slide, and on the slide is your sample. So this diaphragm can be opened and closed, and we can control the intensity or the amount of light which is passing through this lens. And so basically the microscope is a system of lenses. So these are the objective lenses, these are the ocular or the eye lenses. So basically you keep your sample here on the slide, and then with the help of this coarse adjustment knob, you can move the slide up and down. Okay, so when you move this with the coarse adjustment knob, you can move this stage up and down, and you can keep bringing it up. Okay, and keep looking inside the eyepiece. Okay, so once it is the image starts appearing, then you can use the fine focusing to make the image more bright. Okay, so the main trick in microscopy is always focus using the objective lens of the least magnification. So as I said, there are different kinds of objective lenses. In this nose piece there's a 4x which has the least magnification, so that is also called a scanning lens, because when you have less magnification you can see more area on the slide. So field of view is more. So when the magnification is less, field of view is more. So when we increase the magnification, field of view decreases, because image becomes more zoomed. Because as we zoom image, the image becomes big, but we see the less area on the slide. So as the magnification increases, the field of view decreases. So then we have 10x, then we have 40x lenses. So 10x lens can be is also called low power objective, and 40x is called high power objective, and 100x is called the oil immersion lens. So these are the basic parts of the microscope. Okay, so when you are trying to focus some sample, always try 4x first. Okay, because 4x is a scanning lens and you can see more area. Okay, so first focus at 4x and then move from 4 to 10. As the compound light microscope is parfocal, when we change the magnification, the image remains in focus. And then the fine focusing, using a little bit of your fine focusing. So the image stays in focus when we change from 4x to 10x or from 10x to 40x. 40x is also called a high power lens. And then usually in the microscopes which we use in the labs, we also have a 100x lens, and 100x is also called oil immersion lens. We have to use a kind of a oil to use that kind of a lens. Okay, as I said, this microscope is a system of lenses. These objectives, they have different magnifications, and the ocular eyepiece has a um, so objective lenses are of different magnifications, 4x, 10x, 40 and 100. Ocular lenses, they always have only one magnification, which is 10x. Okay, so ocular lens, um, in the monocular microscopes there's only one ocular lens. In the binoculars there are two, but they always have one magnification, which is 10x. So the total magnification is whatever the objective you are using multiply by 10. Suppose you are using 4x scanning lens, that objective has a magnification of 4x, so the total magnification will be 40x. 4x multiplied by 10, so 10 is the magnification of ocular lens. The next objective is 10x. The next objective is 10x, and the ocular lenses, the magnification of ocular lens is 10. So 10 multiplied by 10 is, in total magnification is 100. Okay. So basically, yeah. So basically in this lab, what activity you have to do is you have to just observe some pre-made slides. Just observe them, just focus them, and your observations, you can draw some pictures here, and you are going to submit these pictures on Mercer canvas. That is not graded, but I'm going to see your pictures. Okay, you have to take the quiz, that is the graded item. Okay, now I'm going to quickly tell you about the second experiment we have to do this week. This is called aseptic technique. So first of all, I would like to tell you what aseptic technique is and few more terms, and then I'm going to explain you what activity you're going to do in this lab. And also I would like to tell you, in all these Carolina worksheets, there are very good videos they are given there. They're already embedded. Please watch them. Okay, especially for the aseptic technique, they have made wonderful videos. You must watch them. That's a great demonstration. They'll be very useful to you to learn these basic microbiology skills. So microscope is the very important skill for a microbiologist. And second extremely important skill is the aseptic technique. So first of all, what is aseptic technique? So aseptic technique is the method of handling microbes and material in a way that minimizes contamination. Okay, so contamination is all the other bacteria, because bacteria are everywhere. They can come and they can grow with our culture. So when we are trying to grow some bacteria, we want to have a pure culture. Okay, suppose there are different kinds of bacteria. We are trying to grow E. coli. Okay, and other bacteria which is there in the environment, it starts growing along with E. coli, then that is a contaminant. Okay, we want to grow a pure culture, so that is why we need to use a aseptic technique. So what is sepsis? Sepsis means contamination. A means without. So aseptic technique means handling of the bacteria that minimizes contamination. Okay, we will also need to know two more terms in this lab. Inoculation. So what is inoculation? It's the process of transferring a microbe from one medium to the other one. That's called the inoculation. And what is inoculum? Inoculum is the sample of the microbe which is being transferred. So basically in microbiology, we do the culturing of the bacteria, and for culturing of the bacteria means growing the bacteria in the lab. So for growing the bacteria in the lab, we need to give some medium to the bacteria. Now what is a medium? Medium is basically a nutrition for the microorganisms. It's a nutrition for the microorganisms. So microorganisms, they're life forms. So one of the very important requirement for life is the nutrition, right? So we all need nutrition to survive and to grow and to reproduce. So similarly, microorganisms, they're life forms, they need nutrition. So these mediums, they different as the liquid medium. So if you're taking the lecture part also, you are going over all these things in the lecture part. So the medium can be it can be liquid medium, and in these mediums is basically the nutrition. The nutrients are there on which these microorganisms they grow. So so in this lab, what you have to do, basically in this lab you will learn aseptic technique by reactivating a lyophilized bacterial culture. So in this kit you have been given a lyophilized bacterial culture. So basically it's a bacteria, it's in a dry form. So first of all you are going to reactivate it, and then you are going to use aseptic technique, and then you are going to transfer this bacteria to 3 different mediums. You are going to use a bacterial plate, okay, which is a solid medium, and then you are also going to use a slant. You can grow the bacteria on the slant. And third is the liquid medium, which is a broth. We can also grow the bacteria in the broth. Okay, so that is what you're going to do in the lab. In the lab part, I can show you quickly what you're going to do. So all this material has been given to you. I'm not doing the safety and all those things because that was part of our first lab. The terminology I have already given you. So we are interested in growing the pure culture. So this is the material which you have. This is your like a paraffin candle. So it serves as a Bunsen burner. It basically prevents the contamination. Okay, you have inoculating loops, you have this grease pencil, and you have this rag. Okay, so first of all, the very first thing you are going to do is, this is the lyophilized culture, which means it's a dry culture of the bacteria called Serratia marcescens. So this is the bacteria. So it's in a dry form. So first of all, using a liquid broth, you are going to first of all reactivate it. Okay, so the instructions are great in this worksheet. You have to go over instructions, and there are various videos in this. Please watch them. Okay, so you will be first of all putting some broth into this, and you're going to reactivate it, and you are going to let it sit for 2-3 days, which we are incubating it. So just keep it at room temperature because you do not have the incubator, and let this bacteria grow. And once this bacteria grows, you are going to transfer this bacteria into 3 different kinds of mediums. You are going to transfer it into a plate. So this is a plate. Okay, so this is called this is a nutrient agar plate, and then you have a nutrient broth tube. So this is the nutrient broth tube, and this is the tryptic soy agar slant. Okay, and yeah, so you will be reactivating the culture, and then you're going to then grow it for a few days, and then you are going to transfer to different mediums. How to do that? The videos are already embedded. Please watch them. I'm not going to repeat them. That's just a repetition. And then you will be incubating your cultures. You're going to incubate your cultures, and once your cultures grow, you're going to click the pictures of your cultures, and you are going to post in the observation/activity folder. I already created that folder. Please submit that. I would like to see all your results. Okay, but again, that is not graded. The graded is your quiz. So basically here in this lab you have different activities, and for every lab first of all there's a preparation. You can see that here is a video, inoculation to in this video, where you just come and the video will open and you have to just watch it. Okay, handling the culture tube, it shows you all steps. Even I have created one folder which says a video, in which on the Mercer canvas there are 2 videos which also show you how to use the aseptic technique. Please watch them, and these are very important skills. Okay, how you open the culture, how you take care of the lid, how you use the inoculating loop, how you transfer on a plate, how you run a broth. All these videos are here, and you need to watch them, and then you have to perform the lab, perform the experiment at your home, and you are going to show me the results. You submit the results in the okay. So the very first activity is basically aseptic reactivation of the lyophilized culture. So all these steps are given here. You can go over just one by one. So basically in this I can just overall tell you, I'm not going to go over this each and every step. You can just read it, very straightforward. So you are first of all going to open this bottle, and you are going to put some broth into that, and you're going to reactivate the culture, and then you have to let this culture grow for 24 to 74 hours. Okay, and yeah, so the broth you're going to use this. Remove the cap from the tube of the triptych soy broth. Okay, and you are going to transfer this triptych soy broth to the your lyophilized culture, and you're going to hydrate it. So after rehydration, the next step in reactivation of the lyophilized culture is incubation. So set the tube in the rack and allow it to incubate for 24 to 7 hours. Okay, at or near optimal temperatures, which is printed on the lyophilized culture vial. The culture will be completely reactivated within 24 to 48 hours. Okay, so once your culture is grown, so how to check if your culture is grown? Because your broth will become turbid. Okay, so that shows the bacteria is growing. And after that, you have to transfer the culture from the broth to the broth only. So aseptic transfer from broth culture to broth, and also you are going to do the aseptic transfer from broth culture to slant. And third thing you are going to do is aseptic transfer from broth culture to plate. So all the videos are there, and all the steps are written in a very straightforward manner. Go over those if you have. So at the end, so again, these cultures, you are going to grow them for around 48 hours, and if they're not grown, you can incubate for maybe more time, and then you're going to click the pictures, and then you're going to put them in the folder. So this is the lab, this is the aseptic lab. Basically you have to do, so you have been given a lyophilized culture, you're going to reactivate it, you're going to grow it. For growth you have to incubate it, and then you are going to transfer this culture from the broth to broth. That's number one. Then you're going to transfer from broth to slant. That's number two. And then from broth to the plate. Okay, please watch those videos and do it, and send me the picture and take your quiz. That's all for this week.